Okay, we're going to be making a start on today's lesson. Okay, so welcome everyone to our A Level AQA Biology session. In today's lesson, we are going to be looking at antibodies, antigens, and agglutination. So we've previously done a bit of work on antibodies and antigens in terms of sort of their function, but we've not really zoomed into the structure and we've not looked at agglutination overall. That's what we're going to be looking at in today's lesson, and we're going to be focusing on exam questions, the different types of exam questions that you can get on antibodies. Okay, so if we have a look at the specification just quickly before we start. Now, this is a very broad area of the specification, and we've already had a few lessons on immunity. What we've looked at so far, we've looked at this aspect of it. We've looked at the definition for antigen. Remember, an antigen is a foreign protein which stimulates an immune response. That's your 2 marks that you're looking for. We've looked at phagocytosis. We've looked at the cell-mediated response, and we've looked at the humoral response. So we've looked at all this aspect of it. We've looked at clonal selection. We've not looked at monoclonal antibodies. Monoclonal antibodies is going to be a lesson coming up soon. We have looked at so basically everything your body does to defend yourself against pathogens. What we haven't looked at is the definition of an antibody, antibody structure, the formation of an antigen antibody complex, agglutination. We've looked at plasma cells and memory cells in the secondary immune response. Next lesson, the week after next week will be on vaccination. Next week we're going to do a year 13 topic. We're going to switch between year 12 and year 13 topics, just so if there's anyone in year 13 that would like to do a year 13 topic, they can come every alternate week. If you're in year 12, you can still join our year 13 lessons. You will learn something new from it. But today we're going to be looking at antibodies and antigens, these 3 bullet points, and we're going to be focusing on exam questions. Okay, now the first two, well, over here, the definition for an antibody, and over here, the definition for an antigen. We're going to be looking at that in the format of an exam question. Okay, so I've not given you a definition. I've literally got 2 exam questions that have been asked previously on past papers, and I've put the mark scheme answer. So the question was, what is an antigen for 2 marks? And then the next part of the question was, what is an antibody for 2 marks? So in terms of the definition that you need to be familiar with, the definition that you need to know, that you need to write down in your notes, that you need to memorize is this definition. And I'm not going to give you any other form of the definition. This is literally the only definition that you need to be aware of. Okay, so what is an antigen? We've looked at it before. An antigen is a foreign protein which stimulates an immune response. So if we think about it, your body cells have antigens. Now your immune cells know your body cells. They recognize the antigens on your normal body cells, and they're not going to attack your normal body cells because they recognize those antigens. Now, if anything foreign comes in, so from a bacteria or from a virus, your body is going to detect that as foreign. It's going to look at those antigens and it's going to detect those antigens as foreign, and then it's going to stimulate the immune response. Okay, so those are your 2 marking points. A foreign protein which stimulates an immune response. Now, what is an antibody? That's what we're looking at in today's lesson. So in terms of the antibody that you need to be aware of, an antibody is also a protein, just like an antigen. An antigen is a foreign protein, but an antibody is a protein. Okay, that's how antibodies are made. They are made from proteins. Okay, so that's the first mark by saying that it's a protein, or you could also say that it's an immunoglobulin, a globulin specific to an antigen. Okay, because an antibody, if we think of an enzyme binding to its substrate, an antibody is something like this, that it's binding to its antigen. Okay, so an antibody is also an example of a protein, and it's specific to an antigen. Okay, it's produced by B cells. Okay, so remember when we looked at our humoral response, which is involving B cells are the ones which produce your antibodies. They produce your plasma cells, they produce your memory B cells. Okay, so those are the 2 definitions that you need to be aware of in terms of the definitions and the mark scheme definitions that you need to be aware of. In terms of the exam board that this is taken from specifically AQA. these definitions. However, the definition for an antigen and antibody will be the same across all examples, just the wording might be different. This wording is specific to AQA. Okay, so what we're going to move on to now is antibodies, looking at the structure of antibodies in a little bit more detail. Okay, so when we look at an antibody, we know that an antibody is a type of protein. Okay, it's a type of protein which is produced by B cells and it's going to bind to antigens to stimulate an immune response. Now, an antibody, if you think back to the type of structures that you have, it's an example of a quaternary structure. Okay, it's a quaternary structure protein, which means that it has 4 polypeptide chains. Okay, that's what an antibody is. It's a quaternary structure protein. Okay, it has 2 short chains which are present on the outside. It has 2 long chains which are present on the inside. It has a heavy chain and it has a light chain. Now, if I show you the structure of an antibody, this is what you would see in a structure of an antibody. As you can see, on the outside, you have 2 shorter chains. Okay, so these are your 2 shorter chains over here. This one and this one. Those are your 2 shorter chains. And then in the middle, in the inside of it, you have 2 longer chains. So you can see these 2 longer chains which are going across in the center of the antibody. Okay, because there are 4 polypeptide chains, remember polypeptide chain is just a protein chain. Because there are 4 polypeptide chain, that's what makes it a quaternary structure protein. Okay, now there are 2 regions on this antibody. You have your variable region and you have your constant region. And that's what we're going to be looking at on the next slide. But the main takeaway from this slide is that it's a quaternary structure protein. It has 4 polypeptide chains. It has a heavier chain and a lighter chain. The longer chain, which is on the inside, is your heavier chain, and then the 2 shorter chains on the outside, they're your lighter chain. There's 4 chains, so it's a quaternary structure. Now, in terms of the regions of the antibody that you need to be aware of, there are 2 regions. Okay, you have your variable region and you have your constant region. Now, the variable region is the part of the antibody which is going to be different. Okay, so obviously you're going to have different shaped antibodies with different sites of action, because all antigens are different. All of them are not the same. They are all different, because if all the antigens were the same, if all the antibodies were the same, then they would only be able to attack one type of antigen. Okay, there are so many different types of antigens, and we need all our antibodies to attack all those different types of antigens that you have. So that's why we have a variable region on our antibodies, so they can attack the different types of antigens which come across, because there are always new diseases coming along. Okay, now the variable region is present at the top. So this region over here is your variable region. That's the part of the antibody which is going to change for different antibodies. Okay, and that region that we have there will be complementary in shape to the antigen. Okay, and just remember, when we're talking about enzymes, we mention active site. When we're talking about antibodies and antigens, we mention an antigen binding site. When you come across this in exam questions, never say active site, because they will literally put down reject active site, because this is not an active site. This is an antigen binding site, because on the antibody, the antigen is going to be binding. And then the constant region is basically this bottom part of the antibody, this red part. This red part is going to be the same for all antibodies. So this is your default structure for an antibody. The only part which is going to change and vary to different antibodies is the top part, the variable region of that antibody, because that's going to be attacking different antigens. Okay, you see sort of different strains of COVID appearing. The reasons why, well, we know that strains always appear due to mutations in the virus. They're constantly occurring resistant strains. And the reason why our body is not immune to it is because our body, when we've been vaccinated, when we've had the sort of the disease and we've produced antibodies, the antibodies, they have been produced against the variable region. Okay, against that specific region that we have at the top. Now, when the antigens vary, when the disease varies, when the virus mutates, the antigen shape is going to change shape. And if it changes shape, it is no longer complementary to the antigen binding site of the antibody. And if it's no longer complementary in shape, therefore the antigen antibody complex cannot be formed. When we're thinking about biology, always we're talking about complexes being formed. And the name of the complex which is being formed is always, it's always going to be the first name followed by the second name, complex. So we're forming a complex between an antibody and an antigen. So you're forming an antigen antibody complex. That's the name of the complex that you would say. Okay, so again, the key takeaway from this slide over here is that there are two regions. You have your variable region and you have your constant region. The variable region is at the top. You could be asked in an exam question to mark on a diagram given an antibody where the variable region is and where the constant region is. So you need to be aware of where they are in terms of antibodies and antigens. Okay. So far we've looked at the structure of an antibody. It has a quaternary structure. There are two longer chains and there are two lighter chains. The longer chain is on the inside and the shorter chain is on the outside. You have four chains in total. That's what makes it a quaternary structure protein. In terms of an antibody, there are two regions. You have your variable region and you have your constant region. Okay, the variable region is at the top. That's where your antigen is going to be binding to, and that's going to vary for different antigens because your body needs to have antibodies produced for different antigens. And then you finally have your constant region, which is at the bottom. That's your default structure of your antibody that you're going to have. Okay, now what we're going to do now, before we move on to exam questions, is we're just going to be looking at agglutination. Okay, now agglutination is a really important concept, and in order for all sort of processes, biological processes, in order for the immune system to function effectively, agglutination needs to occur. Let's say a virus has entered your body. Now, because the virus has entered your body needs to detect that that virus has entered your body. If it can't detect that the virus has entered your body, then it's not going to trigger an immune response. Okay, so it's essential that the body detects a virus has entered. And how does it detect it? If one white blood cell detects that virus, that's not going to trigger a whole body immune response. Okay, there has to be a couple of white blood cells that together detect that the virus particles. So what's going to happen? This is the purpose of agglutination. Antigens, they will bind to the antibody, forming the antigen antibody complex. That's a key phrase that you need to use for your exams. Antibodies, they are flexible, which means that they can bind to multiple antigens and they can clump together. And I'll show you an image of this. And because of that taking place, it makes it easier for phagocytes to locate and destroy the pathogen. Okay, so if I show you this image over here, this is an example of agglutination taking place. So we know that the top part of an antibody, that's your variable region. That's the region which is going to change. Now, once that antibody has bound to an antigen, so as you can see, antibody has bound to an antigen, multiple different antibodies can come in and they can also bind to the antigen. Now, once multiple antibodies can bind to the antigen, they form like a loopy network. They form like a loopy network like this, and they basically clump together. Okay, you've got loads of antibodies all in one space. You've got more antibodies than you have antigen or virus particles present. Now, because of that, it's basically like saying, we know, like, let's say if you fall down and you stop bleeding from your knee, blood is going to be pouring out and your body is immediately going to recruit platelets to that aspect where you have sort of falling down to prevent any more blood from leaking out, because that is a big action point. That's basically where all the blood is leaving your body. Just like that, these antibodies, they clump together to trigger a system where all the white blood cells will think, oh, something's big happened here, we need to go and sort it out. That's the purpose of agglutination. So it makes phagocytosis easier to occur. Okay, because when you've got something big happening at a specific site of action, then that will recruit more and more phagocytes much easily. Okay, that's the purpose of agglutination. It's to make phagocytosis occur much easily, and therefore your immune response to occur much more easily within your body. So we know the antigens, they're going to bind to your antibody, and they're going to form your antigen antibody complex. They're going to be flexible, they're going to bind to multiple antigens. So you have more antibodies than you have virus particles or antigens. Okay, and that means that it's easier for the phagocytes to come in and detect the pathogen, and they can destroy it, they can engulf it, and they can destroy it as well. Okay, so that's what's actually happening.